There we go. Today we're going to wrap up Unit 1 in the lab. We're going to take a look at cells and mitosis and cell division, and then we're going to do the cheek cell slides, which we've had great success with all week. Considering we haven't done it for 3 years, I'm very pleased how well it's been coming out, because you'll get a chance to see what your own cells look like. So, and then we're going to go into the skull. I've got the skulls out for you. If you don't have a skull in front of you, there's some extra ones floating around there. They're not real, they're models. So you don't have to have any qualms about touching them. So anyway, let me go ahead and get us into the section here. Everybody that's online should see the slide that says cell cycle and mitosis, and that's where I'm going to open up on. And remind me before we're done to show you where the study guides are for the lab test for next week. It's in the content section of eLearn, but I want to show it to you and walk you through it. So, okay, we're going to take a look at the cell cycle and mitosis. We know that cells are the smallest living structure. Nothing smaller than a cell is alive. Whether you're a bacteria or a fungus, you're still made up of cells. Viruses are not alive. Viruses are non-living pathogens, so they don't count. But every living thing is made up of cells. Whether you're a plant, an animal, a human, a dog, a cat, a fish, a tree, a shrub, whatever, we're all made of cells. The cells are all specialized. They do different things for us. We've gone through all the tissues. We have things like simple columnar cells to absorb nutrients from our small intestine. We've got neurons to carry information for us. We have muscle cells that can contract. Some of the cells are very, very tiny, and some are very, very long. For example, our nerves. Our nerves are made up of individual neurons, the nerve cells. And our nerves, for example, the longest nerve in our body is the sciatic nerve. It starts here at the hip, runs down your leg, behind your knee, goes all the way to ends up behind your great toe. That can be 3 or 4 feet in length. And that nerve is about the size of your thumb. And it's made up of thousands and thousands and thousands of individual nerve cells. So that each cell runs at 3 to 4 foot length. However long your leg is however long the nerve's going to be to get that nerve down all the way to your big toe. The sciatic nerve, because it's so large and so big, does cause lots of problems for people later in life. It's responsible for causing pain, and it can cause pain in the foot, pain in the hip, pain behind the knee, depending on where it gets inflamed. So cells do different things for us. Red cells carry oxygen. White cells fight infections. Cell fragments like platelets stop bleeding. We use, as I said, muscle cells contract, nerve cells send impulses. We use cells to generate proteins. We use cells to break down carbohydrates and release energy. They all do different things for us. One thing about this, every living cell, though, is that they have 3 things in common. If you're a cell, you have 3 things. You all have a nucleus, you all have a cell membrane, and you all have cytoplasm. Now, the only cell that doesn't, there's always an exception. Red blood cells do not have a nucleus. Red blood cells kick out their DNA, their nucleus. They kick out their nucleus before they finish forming, because they want to have more room to carry oxygen, to carry hemoglobin, to carry oxygen, which means that red cells can't reproduce. Red cells cannot undergo mitosis and cell division. Red cells will live for about 4 months, and then they get recycled through the spleen. all the other cells in our bodies are going to have a nucleus, and as well as the cell membrane and the cytoplasm. Today, you're going to be able to see the nucleus of your cells, the cell membrane, and the cytoplasm. Now, every cell that has a nucleus, the nucleus is identical. All the DNA found in a cheek cell is identical to the DNA found in a muscle cell, or a nerve cell, or an adipose cell, or a bone cell. The DNA is all the same. It's just some parts of it are turned on and some are turned off. A bone cell, a living bone cell, is going to have the sections that control how the cell functions, how it gets its nutrients, how it sends signals, how it lays down the bony matrix. But it still has all the other instructions there too, on how to be a nerve cell, or how to be a muscle cell, or how to be an epithelial cell. It's just not using it. So you got to look at our DNA and our nucleus. The DNA is like a cookbook, real big, thick cookbook. If you're going to go follow a recipe to make biscuits, you're going to go to the page where the recipe is for biscuits. You're not going to be following you're not going to look at the instructions on how to, I don't know, make, how to cook a steak, if you will, how to grill a steak. You're not going to look at the instructions on how to grill a steak, or make a pie, or make some other exotic dessert, for example. I'm really in over my head now when I'm getting into a cookbook. However, the instructions are all there, but we're not going to use the instructions on how to bake a pie, or how to make a pie, or grill a steak, or make some specialty dessert, or make shrimp cocktail. We're going to follow the instructions on how to make a biscuit. We don't need to use all the other instructions, and that's what the cell's like. Every cell is going to use its DNA to control its functions. All the other DNA that's in there is still there, but it's not being used. Every cell's nucleus, every cell's DNA is identical. We just have segments of it turned on and turned off. Now, the cell membrane of a cell is the outer covering of the cell. You won't be able to see the membrane, but you'll be able to see where the membrane is located. It's the edge of the cell. The membrane is selective. It lets stuff in and it forces stuff out, but it's very selective. And we have the cytoplasm. That's called the cell sap, the goop, if you will. It has the consistency of hotcake syrup. Very, it's sort of thick and runny, and it suspends all these little structures inside the cytoplasm called organelles that do things for us. Like organelles, we specialize in organelles that will break down carbohydrates to release energy, or we'll have ones that are specialized in how to make proteins for us, or we'll have organelles that fight infections. And these are, they're all very specialized. Some cells have lots of infection fighters. Some cells have lots of mitochondria, that's the energy releasers. Some cells make lots of protein. Depends on what their location is and what they're supposed to be doing. But every cell, except for those red cells, are going to all have a nucleus, a cell membrane, and the cytoplasm. The nucleus is our control center. That's where our DNA is found. It's where our DNA is found in the form of chromosomes. We have 23 pairs of chromosomes in all of our what we call somatic cells. That's everything except the sex cells. So everything except the eggs and sperm are going to have 23 pairs of chromosomes. You got 23 chromosomes for your mother and 23 chromosomes for your father. That gives us 23 pairs, or 46 chromosomes. They are generally invisible when we look at a cell under the microscope, unless they're going through mitosis, because the DNA unwinds. The DNA strands that make up our chromosomes are about 6 feet long, and they about like this long from there to the floor, and they have the consistency of a very, very, very thin thread, a very fragile thread, and it is it's almost like dealing with monofilament fishing line, very, very transparent. It's hard to it's hard to imagine that you've got all of our information is contained in these very thin strands, so that we have 23 pairs of chromosomes, and that contains all of our genetic information. Things like hair color, eye color, curly hair, wavy hair, smooth hair, straight hair, short hair. Is do we have blue eyes, brown eyes, black eyes, gray eyes, hazel eyes? Do we have dangling earlobes, or are they attached? That's a genetic trait, by the way. Can we curl our tongue or not? Curling our tongue is a genetic trait. It's a dominant trait, believe it or not. So if you can curl your tongue, it's a dominant trait. This is a good one. Our DNA controls whether or not when you put your hands together like this, if your left thumb goes over your right thumb, that's a dominant trait. If it's the other way, it's a recessive trait. What does that have to do with anything? But that's controlled by our DNA. Our DNA tells us how to make hemoglobin. It tells us how to make all the proteins we need in our bodies. It tells us how to build our bones. It tells us how to keep our muscles functioning. So all the information we need to function is found in the DNA in all the cells. So the only time we see our chromosomes is when when the cell gets ready to copy itself and the chromosomes condense. Until they're condensed, they are just they're strung out in these 6-foot long strands. So our living cells run through a very specific cycle. We call it cleverly the cell cycle. They've thought long and hard, what would that mean? Anyway, the cell cycle. Cell cycle is mitosis and interphase. Now, at some point, I can remember being told that the stages of mitosis are interphase, prophase, metaphase, anaphase, and telophase, but interphase is not part of mitosis. Interphase is what a cell does when it's being a cell, doing cell things. If you're a mucus gland cell, you're secreting mucus. If you are a columnar cell, you're letting nutrients pass through you. If you are a neuron, you're sending signals back and forth, electrical signals. If you're a muscle cell, you're contracting. We're doing cell things. So 99% of the cell cycle is spent in interphase doing cell stuff. Now, mitosis is the process of replicating DNA. Because when a cell wears out, and cells do, when a cell wears out, most cells will produce 2 daughter cells. They'll undergo mitosis and produce 2 daughter cells. Now, the reason they do that is so we can generate 2 brand new healthy cells with essentially brand new DNA. It's not brand new DNA. It's part of the old DNA and new DNA together, but it's a brand new cell. So, but that whole process of making a new cell only takes about 1% of the cell's lifetime. And if a cell lives for 24 hours, and some do, the last 5 seconds of its life is going to be spent replicating itself. So, question?
So like if you get like a cut and it's trying to heal, is that the mitosis?
Right, exactly. When you have an injury, your body heals it. You generate new skin cells, new dermal cells and stuff like that, maybe new adipose tissue to fill in the site of the injury. So yeah, that's what it's all about. So the cell cycle, yes.
Is this also where cancer would start?
Exactly, exactly. Cancer cells don't respect the cell cycle because the cell cycle has a lot of checkpoints built into it. We're going to talk about that in lecture. But the cell cycle protects us. The cell cycle won't run and make copies of a cell unless everything is perfect. And cancer cells don't care. Cancer cells will just continue to churn out new cells and new cells and new cells again and again. They don't have contact inhibition. They don't care how they grow. So when the cell cycle malfunctions is when we get a cancer. So, and we'll talk a lot about that in lecture when we get into chapter 3. We do know that 2 sets of tissues don't reproduce. Muscle cells. Muscle cells, you are born with all the muscle cells you're ever going to have. They will get bigger and stronger, or they will get smaller and weaker over your lifetime, but you're never going to grow new muscle cells. We have what we're ever going to get. We can exercise and work out, and we can make our skeletal muscles very big and powerful, or we can choose not to, or we can just sort of maintain. Yeah.
So when we're born, it stays the same amount of muscle cells.
Same muscle cells we have now are what we were born with. So we may have lost some over the years as they've worn out, but 99% or so of the muscle cells we were born with are what we have now. So because we don't grow new ones. So, but we can make them big, we can make them stronger, and they will grow in size as we grow, because obviously we don't want to have a muscle cell in our arm that was the size it was when we were little. But when we reach our adult stature, our muscle cells have grown to the adult stature. We just don't make any new ones. So same thing applies to our nerve cells. The nerve cells we have when we're born are what we're going to get. Now, the advantages of all this is we don't have to worry about any kind of errors in reproduction on these cells. And we have enough cells laid down when we were in utero that even if the cells wear out, we still have thousands more, tens of thousands more to make up for anything we might lose. Because cells do age, and sometimes, nerve cells get old. We don't notice it because we've got so many, and we've got like the largest nerve in our body is the sciatic nerve. It's about the size of your thumb, and it's made up of hundreds of thousands of neurons. And if a few thousand of those wear out, you still got hundreds of thousands more to take their place. So now we can repair nerves, not in the brain or in the spinal cord, but in the nerves that feed into the brain and spinal cord. If the nerve gets damaged, we can repair it. The axon, and I think I mentioned this last week, the axon of a nerve, the part of the nerve of a neuron that carries information. If it gets damaged, it can be repaired. Because if we couldn't repair it, then we'd never be able to do any kind of transplant. We'd never be able to reattach a limb. We'd never be able to put a new face on somebody, or transplant an organ like a kidney or a liver or a heart, or a lung, because we have to connect the axons together, and they'll grow back and they'll grow together and function. If we couldn't do that, then what's the point? So unfortunately, we can't do anything like that in the brain or spinal cord, but we can do it in the nerves that are outside that. So, okay, our cell cycle then is 99% interphase, followed by the stages of mitosis. Now, what the cell cycle does during interphase, it's busy doing cell stuff. But at some point in the cell cycle, it decides, I'm getting old. It's time for me to create daughter cells, and it will make a complete copy of its DNA. Because mitosis is what we say nuclear replication. We copy the DNA. We make another set of the DNA. So for a very, very short period of time, our cell has 2 sets of DNA. Just sitting there ready to go. And this is a very, very brief period of time because we don't want to have 2 sets of instructions in the cell. So, Yes. Okay, so we're ready to go. During interphase, we make a complete set of our DNA. And now we're ready to go ahead and start the process of mitosis. You know, this mitosis consists of the stages we probably all heard about: prophase, anaphase, metaphase, and telophase. The whole purpose of mitosis is to be able to replicate our DNA and put two perfect copies of our DNA into daughter cells. Depends on how long the cell is going to live. You know, some cells live forever. You know, well, not forever, but they live our lifetime. They don't ever copy. Some cells last for a day. Some cells last for a few days. Some white blood cells, for example, the infection fighters, show up right away and they're good for about 24 hours. We keep making more and more of the same type. Other types of white blood cells will last a lifetime. They're the ones that give us immunity over time. They create they're the antibody producers that fight infections. So that, you know, if you have immunity to a certain disease, every time you get exposed to it, you don't even become symptomatic, you don't get sick because of these white cells. And so you create a library of white cells that will fight infection. So anyway, this is interphase. Cell, you know, the nucleus is visible, very, very clear right there, the big purple thing. You can't see the chromosomes, you can't see anything other than some dark splotches in there, and that's okay. The chromosomes are unwound. We can see the cell membrane, the edges of the membrane, and we can see the cytoplasm. We can't see the organelles in there, but they're there. We want to get into mitosis. As I said, the last 1% or even less of the cell cycle is all about mitosis. Mitosis is the way that we can take two sets of DNA and evenly distribute them into two new daughter cells. And this shows you just how fast that takes place. This is real time of video of the process of mitosis, going through the 4 stages of prophase, anaphase, metaphase, and telophase. That one there in particular I really like because that's showing the stage of anaphase where the chromosomes have condensed and start pulling apart. It's the shortest of all the stages. We also see how the cell then divides at the end of mitosis and how quickly it happens too. You know, most textbooks describe mitosis as, you know, a very long section of the cell cycle, and it's not. You can see how long it takes to give you daughter cells. Okay. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. In prophase, the chromosomes condense. Chromosome condenses and they pair up. Because you've got 23 pairs of chromosomes that have copied themselves. And so in the 23 pairs, you've got a pair of chromosome 1s. You've got a pair of chromosome 2s and a pair of chromosome 3s, and so on until you get all 23 pairs of chromosomes. The 23rd pair are the sex chromosomes. It's going to either be an X and a Y or an X and an X, depending on the sex of the individual. They condense, they become visible. The nucleus, the nuclear membrane breaks apart and disappears. You have the chromosomes start to migrate towards the middle of the cell, and we see something called spindle fibers start to form. Spindle fibers are fibers that are going to go, and some are going to attach to the chromosomes to pull them or push them to opposite ends of the cell, and others are just going to be pushing on the cytoplasm to push it around too. So this is prophase, very quick, very brief phase. Metaphase. Metaphase is probably the easiest one to spot the chromosomes line up in the middle of the cell. And in metaphase, the reason they do this is so that we can have equal distribution. Because remember, you've got two sets of DNA. So you got to line up to make sure that when the cell does undergo division, that both daughter cells are going to have, each one's going to have 23 pairs of chromosomes. And the way to do this is to line up along the middle. Oh, and did I tell you that the reason why the chromosomes condense? Because it's a whole lot easier to move a stubby little chromosome where it's all wrapped up on itself than it is to try and move a 6-foot-long strand through the cytoplasm. So otherwise it might get twisted and break apart. Okay. Anaphase, easily the most visible. With anaphase, we see the chromosomes being pulled apart to opposite ends. This is the fastest, the shortest of all the stages of mitosis. We also see what's called cytokinesis starting here. In cytokinesis, it's when we start squeezing the cell in half. We use a protein, the same protein we find in our skeletal muscles. It's a contractile protein, and it starts squeezing the cytoplasm. And it squeezes it. It looks like one of those balloon animals, that you twist around. And only this time it's a balloon animal that when you twist it, it'll separate. And that's what it does. And then the last stage is telophase. In telophase, the chromosomes have moved to opposite ends of the cells. The nuclear membrane starts to reform. Cytokinesis is completed in this stage. And you can see here we have two stages of telophase. Here the cell membrane we haven't completely separated the daughter cells. Here the daughter cells are almost completely separated. Cytokinesis has taken place along here, and we have almost two distinct cells at this point. And we see this one is even further along, forming these daughter cells. We have what's known as the cleavage furrow taking place right along in here as we squeeze them apart. And those cells now will live whatever the lifespan of the parent cell was. If it's 2 days, if it's a week, if it's a month, it just depends. Depends on how long, whatever the average lifespan of that cell was. As I said, some cells will last for days or weeks or months or even years, and some are very, very short. It's been estimated that a cell line, meaning all the cells, you have an initial cell, and it will undergo mitosis and generate 2 daughter cells, and they'll undergo mitosis, generate 2 daughter cells, and so on and so on. It's been estimated that a particular line of cells will go through 50 sets of mitosis, 50 replications before it stops. So you could produce lots of cells doing that. So, okay. And that's cell division and mitosis, very briefly. Okay. Now, the next thing we want to do is take a look at cheek cells. Now, this is a very simple process, and it's worked all week. You take a microscope slide like this. Now, the microscope slide is going to have a white frosted area on it that says specimen. You don't do anything with that. You want to use the clear portion. The reason I'm saying this is because I had people in one of the labs put everything on the white frosted area. So anyway, so you're going to take a toothpick. It's spared no expense in these toothpicks. So they're the best you can get at Walmart. So, and you take the flat end of the toothpick, rub the inside of your cheek, smear it on the glass